All right, so I'm going to talk about plant-derived bioactive molecules, especially novel antifungal agents in next few minutes. And we will just talk about what exactly is the secondary metabolites and what exactly these plant-derived agents are being used uh, in medicine, or how they are going to have pharmacological property. So plant-derived bioactive molecules or plant secondary metabolites are metabolites which are often produced in a phase of subsequent to growth. It has no function in growth and development, and these secondary metabolites are produced by several taxonomic groups of plants and other organisms. They are very diverse structurally, and also present in the mixtures of several closely related chemical structurally similar molecules. In general, we can define secondary metabolites are the metabolites are not required for growth and development of that particular organism, but they are also play very important role in the life cycle of that particular organism. So initially it was considered as these molecules are nothing but the byproducts of some of the metabolic processes. But later on, it was found that these molecules are playing very important role in several processes. For example, just before going into the details, we would like to see the difference between primary and secondary metabolism. So all of us know what does it mean by primary metabolism, right? And then what is secondary metabolism? So primary metabolism basically involves a central metabolic pathway, and that pathway is providing energy for the growth and development of that particular cell in general, and the organisms in particular, right? So in case of plants, primary metabolism includes photosynthesis, respiration, and other metabolic processes or biosynthetic processes. So these metabolic processes provides intermediates for secondary metabolic pathways. So main function of primary metabolism is providing energy and building blocks for the growth and development of that particular cell and organism. But secondary metabolites are produced under specific occasions. Unlike primary metabolites, they are not produced constitutively or each and is what we can say regularly, right? So these secondary metabolites are produced under certain conditions. And what type of secondary metabolism we know so far? Basically, there are three groups of secondary metabolites, that is nitrogen containing alkaloids, phenolics, that includes flavonoids and other phenol containing molecules, and terpenes and terpenoids, the molecules responsible for flavor and fragrance of the aromatic plants. So what is the purpose of this secondary metabolism or metabolic pathways or secondary metabolites? So as you can see in this slide, these secondary metabolites are playing very important role in the though it is not in the growth and development, but it is involved in several processes. Yes, we were talking about the purpose of this particular secondary metabolites and how these secondary metabolites are helping or facilitating several important processes in the life cycle of plant. So as we can see, these secondary metabolites are providing tolerance towards several stresses or stress conditions. So may it be a biotic or abiotic stress, or it may play very important role in defense mechanisms of the plants. So as such, as these molecules are basically produced or synthesized in response to certain stress condition, it may be temperature stress, drought stress, or it may be against the pathogenic microorganisms, maybe against bacteria, against fungi, against viruses, or as against insects and herbivores. So to protect that particular plant from these different stresses, these molecules are synthesized. Basically, they are evolved right through the evolution, and that's how those secondary metabolites have started protecting those plants from the stress condition. And that's why these molecules are highly diverse structurally. So you can get a whole, it can be considered as a storehouse of structurally diverse molecule, and those structural diversity of that particular molecule provides or facilitated or enables that particular molecule to have different pharmacological activities. So there are three, as I mentioned, there are three different groups: nitrogen containing, phenolic compounds, and terpenes. So these are the three major classes of secondary metabolites, and this is how they are synthesized. So you can see the intermediates of different primary metabolic pathways are being used as a precursor for these secondary metabolic pathways or synthesis of these secondary metabolites. Like so, you can see terpene is the largest group of secondary metabolites, and more than 29,000 terpene molecules are described or identified till today. There are more than 8,000 phenolics molecules are known. 12 to 15,000 alkaloids are known today, and in detail, if we go, these terpenoids are further classified into different groups based on the structure: mono, cis, quiterpene, diterpene, triterpene, steroid, sapone, etc. Phenolics are further grouped into flavonoids, polyacetylens, polyketides, phenylpropenoids, etc. And alkaloids are again classified based on their structure and the composition of that particular molecule into different groups. So that's how these plants can provide a very large group of molecules and structurally diverse molecules which can be exploited and which are being exploited from the ages by human beings for different purposes. So we know what exactly we are using it for. So before this modern medicine came into existence, so plants and plant derived products were the lifelines of human beings for protecting or for protecting themselves from the diseases, from not just from human, even for animals also. So how we learn that these plants are going to be useful and they have different pharmacological properties is because it was the observations of our forefathers who used to visit these woods and jungles and see that how exactly these animals and these what we can say all different animals survives under different conditions. And as we know, these birds and animals eat different plants under different stress condition and they survive. And like that, there are many stories and many books are available which has given beautifully the background how we came to know about those plants, and then whole systems, traditional systems of medicine, not just Ayurveda, Siddha, and Unani, but Chinese traditional system of medicine and other traditional systems of medicine all over the world are dependent for medicine and preparations for the from the plants or depended on the plants. So that's how plants have provided a storehouse for different molecules and different drugs for the treatment of various diseases. So how these molecules are going to have this different biological activity and how these molecules show pharmacological properties that we will discuss. But in general, we can just have a brief account of these different groups, terpenes, then different types of terpenes, phenolic compounds, different phenolic compounds, uh, basically part of the plant defense system, uh, produced against diseases, pests, and parasites. And that's how plants protect themselves against these organisms or these pathogens. Alkaloids are the nitrogen containing compounds, and they're very, very interesting molecule. Several alkaloids has been in use as a drug, and since they are structurally diverse, and based on the structure, they are again grouped into different alkaloids. So these are the brief about the plant secondary metabolism or metabolites or plant derived uh, bioactive molecules. So since limitation of time, we cannot go into the details of these different types. But I would like to present a piece of work we are doing in our lab. And what exactly we are doing in our lab is we are looking for novel antifungal agents. So how we are looking towards this plant and how these plants or natural products are facilitating drug discovery. So about 25% of all the prescriptions sold in US are derived from the plant or plant, and another 25% is basically structurally modified natural products, right? So these, and there are several, you can see the list of these drugs basically derived from plant, and they are being used. They are currently available in the market and being used against several diseases. So that this is how these plants are providing a source for novel drug uh, against various diseases. So we know the significance of this particular aspect, drugs, as we have seen whole country, and not just whole country, whole world is locked down because of the lack of effective drugs against COVID-19. And still we are waiting for a drug or a vaccine against this particular virus, and still we are struggling. Nobody knows when we will get the drug or when we will get the vaccine. But yes, sooner or later we get effective drugs against this particular virus. And I hope plant derived agents will fulfill that particular dream, as a lot of plant biologists or phytochemists are working on identifying novel antiviral agents, especially for the COVID-19. So, with this, I would like to uh, proceed towards the work we are doing in our lab and what exactly we did from last few years. So we are working on uh, antifungal agents. It is because, as we know, the antimicrobial resistance has become one of the most serious global issue, and you can see the figures, how many deaths are being caused by this particular antimicrobial resistance. So antimicrobial resistance is nothing but the resistance of that particular pathogens towards specific drugs, and it is a evolutionary process. So as long as the particular organism is exposed to the antibiotic or antimicrobial agent, slowly it is going to develop resistance towards this particular drug. And that's how we need to keep searching for novel antimicrobial agents. And it is a constant interplay between microorganism and the human or host body. And that's how this particular antimicrobial resistance has become a serious global issue. So uh, we people are pouring money into the research of those effective antimicrobial agents, but still there are no promising antibiotics available in the pipeline. And with respect to the antifungal agents, we are restricted to only few groups of compound, that is azole, echinocandin, and polines. Sorry for the typographic mistake. It is not Polynesia, it is Pauline. So these are some of the groups which are found effective against fungal pathogen. And uh, unfortunately, though azole and echinocandin and pollines are known for their efficacy, recent data shows that most of the fungal pathogen have started evolving resistance towards these drugs, especially against azole, because Pauline, though it is effective, it has severe side effects and it has restricted use of that particular drug, and it is only being used under the critical or conditions or terminally ill patients only. It is not the routinely used drug, Pauline class of drugs. So azole was the drug of choice against fungal pathogen, but since uh, resistance toward this azole is evolving against several fungal agents, it has again limited use of azoles, and that's why the search for novel antifungal agents are being searched, or people are trying to look for the novel antifungal agents. So this is in brief about the antifungal agents, their mechanism, how exactly they are acting, what are their targets, and why we need different or novel antifungal agents. So we did some work on novel these plant molecules. We screened several plant molecules, identified novel antimicrobial agents, and we screened several essential oils against the fungal growth, right? And later, that study was focused or zeroed on the effective molecules, which was found to be promising in our earlier studies. So we tried to understand how exactly these molecules are inhibiting growth of these fungal pathogens, especially Candida albicans. So we have tested efficacy of different plant molecules, especially the molecules derived from the terpene or terpene class of compounds or terpenoids, and we were looking for the effective terpene molecules against um, these Candida albicans or fungal agents, based on our earlier study that essential oils have shown promising results against the fungal growth. And as these essential oils are nothing but the mixtures of different terpenes and terpenoids, or we can say isopenoids, we decided to check or to identify the major bioactive principle of that particular essential oil, and that's why we have tested these different components of essential oils individually, and not just that efficacy, we have tested or we have evaluated mechanism or mode of action of those particular molecule against fungi, how these molecules are inhibiting fungal growth. So we tested effect of these molecules on uh, planktonic growth as well as different form of growth of these fungal pathogens, planktonic growth, biofilm growth, and mycelial or hyphal form growth as well. And these are the data of some of the molecules. So we have determined MIC. MIC is nothing but the minimum inhibitory concentration of that particular plant molecule against the growth of that particular pathogen. And you can see there are differential efficacy exhibited by these particular compounds, and It is not only osmium, cinnamaldehyde, farnesene, curcumin, and campane. We have tested several other molecules. This is just for representative slide. We have tested effect of these molecules, right? Combine effect, right? So different combinations we tried with the known antifungal agents, and we found that these molecules, right, act synergistically against growth of this fungal pathogen, not only planktonic but even biofilm growth. So we have used one organism which is resistant to fluconazole, but when we use this fluconazole along with the molecule, subtoxic concentration of this osimine, farnesene, and other molecule used in the study, we found that subtoxic concentration inhibited fungal growth significantly, and it uh, based on that result, we concluded that these terpenes we have tested or we have evaluated in our study exhibits excellent synergistic potential against fungal growth. So you can see here how we did MIC and MFC of these different molecules. You can those with the on plate AC, we sprayed specific number of candalbecan cells on the agar these YPD agar plates. It was grown for um, 24 hours, and then you can see those colonies. So the concentration which is inhibitory or which is responsible for killing 100% inoculum are considered as minimum fungicidal concentration, and concentration was inhibiting growth but failed to kill the 99.9% inoculum are considered as minimum inhibitory concentration. So difference between MIC and MFC one should know very clearly. In addition to MIC and MFC, we also did the kill curve assay to determine how much time that molecule need or that organism need to be exposed to the MFC of that particular molecule, right, for killing 99.9% inoculum. And you can see this particular graph here. So different molecules need different time for killing, and that's how you can osimine requires only 20 minutes exposure, while cinnamaldehyde require 80 minutes and osimine require 20 minutes of exposure, and within 20 and 80 minutes these minimum fungicidal concentration of osimine and cinnamaldehyde can kill 99.9% of inoculum, and that's how we determine the effect of this molecule against biofilm growth. You can see different concentration and you can see the growth of that biofilm at the 24 hours and 48 hours. So this is the biofilm growth after 48 hours at zero concentration, and different again 12 and 48 concentration in case of 48 millimole concentration in case of campane, 15 and 60 millimole concentration of curcumin, 75 and 300 millimole concentration of farnesene, 1.25 and 5 millimole concentration of cinnamaldehyde, and that's how these concentration can inhibit the biofilm formation. Now we were wondering, since these molecules are inhibiting the fungal growth significantly or very effectively, how exactly these molecules exert antifungal effect, how they kill fungal cells. So that was the quest, and then we wanted to understand that mechanism of action of those particular molecules, and we did that study in detail. This is different molecules, and that study was published in different journals. So this one paper was published in Medical Mycology, and this year also we got one more publication on piperine in the same journal. In addition to that, we got some paper on menthol and some other molecules. So we understood the mechanism of action of these molecules against fungal pathogens. So how we did that? So we tested effect of these molecules on planktonic and biofilm growth. Then we tested effect of that molecule on morphogenesis, and we can see that these molecules can inhibit the budding, or this inhibit hyphal formation. So as those who know about the kind albicans, that kind albicans exist in different morphological form like hyphae, pseudohyphae, budded and unbudded cells. So when you induce that particular hyphal form growth, right, in presence of this natural molecule or these compounds, plant-derived agents, it has inhibited that hyphal growth, and that's how instead of hyphal form growth, that organism started growing in this budded and unbudded form. So because hyphal form is considered as highly pathogenic, more dangerous than the yeast form growth, because it invades tissue and cause invasive infections that is more dangerous than the superficial candidiasis. We have tested synergistic effect of limonene along with the fluconazole, and then we found that only zero point 0.6 millimole of limonene and 0.02 milligram of fluconazole can kill, sorry, 0.01 millimole of fluconazole and zero point 0.6 millimole of limonene can inhibit Candida albicans growth completely. Where is on the other hand, when we use individually, so that fluconazole concentration was needed at very high concentration, right? But in case of combination, very less concentration was enough to inhibit the planktonic and biofilm growth of Candida albicans. So in addition to this growth inhibition, we decided to understand how exactly this limonene is inducing the or inhibiting the growth of Candida albicans, and we identified these limonene responsive proteins, right, of the Candida albicans using LCMS data, and we found that these proteins are modulated, right, down regulated proteins and upregulated proteins were belonging to different metabolic processes like transcription, translation, mitochondrial function, metabolism, proteolysis, and many cell cycle, etc. So these proteins were modulated in response to limonene, and to support that particular data, we did some cell cycle assay, some microscopic assay, and we found that this limonene is inducing apoptosis in Candida albicans. You can see this image shown in the upper side, E2, right, where you can see the nuclear, right, fragmentation in the nuclear compared to the control cells, where you can see intact nuclear. And that's how we concluded that this particular limonene is inducing apoptosis in the cell. And how this is inducing apoptosis, we further confirmed it using the flow cytometric analysis. And flow cytometric analysis has also shown that limonene is inducing apoptosis in Candida albicans. So we were wondering how this limonene is inducing apoptosis. How exactly? What is the effect, or how? What exactly is the mechanism? So we concluded that this limonene is inducing cell wall and membrane damage, or causing cell wall and membrane damage, and that cell wall and membrane damage is upregulating or inducing production of reactive oxygen species. And to cope up with those reactive oxygen species, it has impacted several genes, and we found that genes involved in managing that reactive oxygen species stress, there are some of the genes, they were upregulated, while some of the genes were down-regulated. And we also found that the induction of calcineurin pathway and also the nuclear stress mechanism. So based on this overexpression of some of these proteins in response to limonene, we could link that particular mechanism, and we propose this particular model of the mechanism of limonene induced apoptosis in canine albicans. And this model was further supported by expression of those selected genes, like we checked the expression of ERG11, MCA1, which is involved in apoptosis, KRE9, which is involved in membrane and cell wall structure. ERG11 is again involved in membrane structure, that is sterol biosynthesis. TRR1 is involved in the oxidative stress, and RPL11 was involved in nuclear stress. And overexpression of these genes in response to limonene supported the hypothesis that limonene is inducing the cell wall and membrane damage stress that induces the oxidative stress, and oxidative stress followed by the apoptotic signals, that is overexpression of MCA1 and this expression of RPL11 that is responsible for nuclear fragmentation, or that is a marker of nuclear fragmentation. So this is how we checked and we evaluated the mechanism of action of this particular limonene. And this limonene is nothing but a component of the essential oil that we already tested against canine albicans. So initially we tested that essential oils and then looked for the different components of that essential oils for the antifungal activity. And we found that limonene was one of the most effective molecule, and we tried to understand how that limonene is inhibiting fungal growth. And this is the hypothesis, and this is the model we propose, that limonene is inducing cell wall and membrane damage, reactive oxygen species, followed by DNA damage and nuclear stress, and that's how finally it is inducing the apoptosis. So this is the story of limonene. Similarly, we have tested effect of evaluated effect of menthol. Menthol is one of the most important and most frequently used terpene, and it is used in several even drug industry, even chocolate industry, even in tobacco industry. So this menthol, and it has several pharmacological properties, and this menthol is the major active principle or ingredient of the mentha essential oil of the this one, and that's why we decided to check the efficacy of that particular molecule against the growth of canine albicans. In addition to menthol, we also tested effect of piperine against the growth and development of this particular organism and different morphological form growth, right? Like yeast hyphal form growth or biofilm form growth or planktonic growth. And not just that, we again identified these menthol and piperine responsive proteins using LCMS analysis. And based on our observation, we propose that mainly these terpenes or terpene-like molecules are membrane-active molecules, and these molecules are inducing membrane stress, membrane damage stress, and that stress further induces reactive oxygen species, and that reactive oxygen species is further induces apoptosis in canine albicans. So mechanism of this limonene and menthol and even in piperine and curcumin are through the damaging membrane and cell wall of the canine albicans and finally inducing apoptosis. So that's how these plant-derived agents are inducing the apoptosis, and that's how inhibiting the growth of fungal pathogens.